attention today, and welcome to the Trifoil Imaging AXT webinar on advances in optical imaging, new probes and instruments which make full 3D in vivo imaging a reality. My name is Steve Hunter. I am the Business Development Manager for AXT's and vivo imaging products. For anyone unfamiliar with AXT, we are Australia's leading high-end scientific instrumentation supplier with clients all through the Australia and New Zealand region. We represent almost 50 manufacturers and sell products into the life sciences, material science, mining and industrial testing sectors. With regards to our in vivo imaging products before now, you can find out more information on the AXT website at www.axt.com.au, or feel free to contact me directly. Of all our products in this portfolio, the Trifoil Imaging FLECT is probably the most exciting and innovative, as it allows translational imaging by means of optical imaging. Let me explain. The gold standard for translational molecular imaging is PET, positron emission tomography. And usually that's coupled with either CT or MRI for an anatomical reference. However, excuse me, this technology is often beyond the reach of most smaller labs and institutes because of the cost of the instrumentation and the difficulty with radiation licensing and even getting access to those isotopes. And I believe that in this space, this is where the FLECT CT fills this gap in terms of its capabilities, and allows smaller labs and institutes to pursue translatable medical projects. Of all the imaging modalities, molecular optical imaging is the most common technique for in vivo imaging of small animals, but it does suffer from a lack of ability to quantify and also with resolution. So essentially there's no tomographic capability with these technologies. The FLECT CT, however, provides these capabilities and allows these techniques to be done on a much more affordable budget. Today's presentation will be given by Dr. Wesley Moy from Trifoil Imaging. He's a Director of Technology Development, and Wesley has studied at the University of California, Irvine, where he obtained a Masters of Science and a PhD in Biomedical Engineering. He's had extensive experience with laser-based techniques for microvascular treatments and also optical imaging techniques for studying respiratory disease. He's been with Trifoil now since 2017. And I'm sure you'll find his presentation today very insightful, and no doubt it will open your imagination to some new possibilities. If you have any questions during the presentation, I'd like to ask you to use the question and answer facility in the bottom of the webinar window, and we'll come back to those questions at the end of the webinar. So before we go into the main presentation, I would like to hand over to Aaron McCormack, who's the CEO of Trifoil Imaging, for a brief introduction to the company, and he'll then move on to Wesley's presentation. So thank you, Aaron. All over to you.
Thank you, Steve. Good morning, everybody. My name's Aaron McCormack, and a few months ago I became the CEO of Trifoil Imaging. It's been a privilege to work with Austin, Wesley and our team here, as well as the good folks in AXT and our customers all over the world. I'm based just outside of Boston, Massachusetts. An Irish guy in Boston. I'm a walking cliche, really. So the crux of this evening's presentation, or this morning's presentation in your case, is to talk about a single orthodoxy that I think is commonly held still across the preclinical research and clinical research community. And that orthodoxy is that optical imaging isn't able to see into the mouse. There are tens of thousands of publications out there which have used optical imaging in the preclinical setting to demonstrate answers to certain scientific questions, but most people, if asked, would still believe that it's not possible to see much deeper than a few millimeters into the mouse, and therefore the whole challenge of optical imaging in orthotopic models and in other things that require seeing deep into the mouse is avoided. And that's most stark actually in the current virology and respiratory disease setting, as we'll talk about later in the presentation, where where it's almost not used at all because people feel they can't see into the animal in the way that they would need to. And I think what we've been trying to say from a trifoil imaging perspective and way back into the bioscan days is what would you do if this was no longer true? How would this enhance your research? And at Trifoil Imaging, we've made that our exclusive mission over the last couple of years to bring true 3D optical imaging to the marketplace and to enable researchers all over the world to make use of it. You may not have heard of us. I know some of you already know us well, but if you haven't heard of us, I think it's important to talk a little bit about who we are. We're not a startup. Actually, we've been around for over 20 years, and our heritage is a little bit of GE Healthcare, a little bit of a company called Gamma Medica. Many of you will be familiar with them, and a good sprinkling of another company called Bioscan. And Bioscan was a company that had a lot of the intellectual property and capability that makes up our flagship instrument today. But as part of our GE Healthcare and Gamma Medica heritage, we support over 275 radionuclear, PET and SPECT, and CT instruments in all the locations you can see on the map here, including in Australia. And I think that's important because if you haven't heard of us, you might think we're just a startup bringing an instrument to market. I think it's important to know that we understand what it is that researchers need to be able to perform their work. We certainly understand radionuclear modalities and what they can deliver to you. And our mission, as I say, exclusively for the last 3 or 4 years, has been to make sure that optical imaging can deliver a full body 3D alternative that can either take the place of radionuclear imaging where it's not possible, or verify it where that is required. So thank you very much for turning up to talk with us today. I'm going to hand over to Dr. Wesley Moy now, and he'll take you through the rest of the presentation. Wesley, over to you.
Thank you, Aaron, and thank you, Steve, for the nice introduction. So today we'll first start by talking about the different choices of modalities that you can choose from when you are starting out with your different research projects. I'll start by going over this figure on the left. So we have different resolution on the bottom and also different imaging modalities on the y-axis. And as you can tell, for specific types of imaging, you can get different types of information. So for example, X-ray CT is good for anatomic or physiological imaging, same with ultrasound. But then up until metabolic or molecular imaging, it's not suited for that. Specifically, we can also look at magnetic and nuclear and optical methods, and we'll focus more on the bottommost, optical, for today's presentation. So where do we begin? What sort of research question are you trying to ask? Is it a confirmation of some phenomena? Is it a detailed analysis? Is it some combination of both? Then the next logical progression is, what instruments are available at the facilities that you're currently working in? And then do you have specific types of probe or chemistry that enable access to these different modalities that then give you the type of information that you're looking for? And in addition to that, you also probably are trying to look at how to clinically translate from benchtop all the way to bedside, as that's what the end goal normally is for most research projects. Other considerations are volume, throughput, or number of animals that you need to actually run your study, the ease of the actual experiment, ease and simplicity, the cost, so cost basis, whether or not you need to use radioisotopes or if you need access to very expensive instrumentation. And then there's also concept of 3 R's of reduction, refinement, and basically trying to minimize the number of studies that you need to do to perform your research. So for multimodality imaging, we specifically focus on both the anatomy and molecular processes, and that's the instrumentation that we have designed and have brought to market, which we'll go into a little more detail later on in this presentation. So how do you image a molecular process? Well, we first start with a targeting molecule, and this is specific to a biomarker or a phenomenon that happens in either a disease model or some kind of progression state. So this could be in the form of a chemical compound, and an example would be glucose, that's overexpressed in the presence of tumors, or an antibody, something like VEGF, vascular endothelial growth factor, or CD44. Then you need to have a method of contrast that allows you to visualize this targeting molecule. And we can use a number of techniques. We can start with radioisotope base, used in PET and SPECT that emit radiation. An example here is FDG18. You can conjugate to a fluorescent dye, something like ICG, indocyanine green, the cyanine families, or an alexylfluorophore family. And then in addition to that, you could also use reporter genes. So these light-emitting proteins, which are typically used in bioluminescence and also fluorescence applications like luciferase, green fluorescent protein, red fluorescent protein, and specifically, which we want to talk about in more depth, infrared fluorescent protein. Lastly, as you guys probably know too, there are also nanoparticles that are available, either the up-converting or down-converting versions with high fluorescence efficiency that can be made to specific applications. An example here would be quantum dots. And there are several others that we can talk about. So this concept of a molecular probe is basically the combination of a targeting molecule plus this method of contrast. And this really ties into these specific aims for molecular imaging, which are in vivo assessment, specific visualization of your molecular target, and then specific localization, or where is it with respect to the anatomy of the subject that you're interrogating. So what we have done is specifically designed our instrument to optimize the visualization and localization while providing a non-invasive method to achieve this goal. So we'll quickly go through this slide, which basically is, there are 6 different image panels here, and what we want you to do is choose from these options below: SPECT, PET, MPI, or magnetic particle imaging, MRI, bioluminescence, and fluorescence. And at the end of the presentation, we'll go over what these answers are. Some of them may be a little bit more obvious than others, but maybe you might have some difficulty depending on, because they look very similar. But we'll get back to this and why this is important later too. So focusing on optical molecular imaging. What is optical imaging? It's use of visible infrared, the visible to near infrared region of the EM spectrum to interrogate biological tissue. And what we have done is specifically focus on the near infrared region because of some tissue optics properties. And we'll go over some basics first with what fluorescence and bioluminescence are. So fluorescence is presence of a molecule that absorbs excitation light, and then this molecule jumps to an excited vibrational state. And during its relaxation back down to the ground state, it emits a photon of light that's longer in wavelength. In bioluminescence, it's an enzyme substrate reaction and specific generation of spontaneous emission of light. And keeping in mind there are different time points that need to be acquired, and also other considerations with each one of these phenomena that provide both pros and cons to why they could be useful in certain applications and not so useful in others. So as I mentioned, there are some advantages. In molecular imaging using optical, we can basically perform this molecular targeting without having to use ionizing radiation. The sensitivity is comparable to PET and SPECT, and as Steve alluded to at the very beginning in the intro of this presentation, the gold standard is PET or SPECT with an anatomic reference. And so we can use an optical analog that can get us similar sensitivity. In addition to with optical imaging, you actually have very good high throughput screening. So the ability to process or image many animals at once and have a large number of cohorts for your study. With fluorescence, there are many agents, many commercially available dyes. There are near-infrared fluorescent proteins and nanoparticles. And from a temporal standpoint, without going too much into other parts of fluorescence, it is consistent with signal. So it's relatively time independent. With bioluminescence, the advantage is there's no background, meaning the signal that you get strictly just only comes from this enzyme substrate reaction. And then finally, there's an economic and effectiveness to answering these basic questions using optical imaging. Some of the challenges, however, are that with most optical imaging systems, they are 2D planar imaging. So it's a fixed camera location with the subject underneath. You lose out in depth information, and quantification can be something that is difficult to perform or inaccurate. And the best example I can give is if you have a signal that's close to the surface, a weak signal that's close to the surface can also be displayed as a strong signal deeper into the animal. And it's very difficult to differentiate where the source is coming from and what the difference is in these two signal types. With fluorescence, there can be a non-specific background that can include autofluorescence or presence of your targeting agent not necessarily going to the specific target and then visualizing the signal in other parts of the body. With bioluminescence, the signal intensity decreases over time. So, as you guys are probably aware of, preaching to the choir, but there is a time dependency on the signal intensity, and as you need to find the optimal imaging window, and it's also one snapshot within a dynamic process that could change metabolically from different animals. So there's a lot of moving parts that come into play with these different imaging types. In fact, finally, the pathway to clinical translation or optical to an isotopic modality, it's difficult because it's hard to take planar images and then convert them to a tomographic format, which is where PET and SPECT are also taken in a tomographic format. So the idea of having a costly and difficult and inconvenient solution in these clinically operated modalities, this is where we can try to bridge this gap using an optical imaging method. So as I mentioned, how do we go from 2D to 3D? And can we do this in a way without jumping straight to a radioisotope-based method where you can get depth information, you can also perform quantification, and you have a direct clinical translational pathway? And so we focus specifically on the near-infrared region of light. And so why did we do that? NIR has some very optimal light tissue interaction properties. This is due to the scattering nature of tissue and also reduction of absorption from these specific chromophores, so water, oxyhemoglobin, and hemoglobin in general. And this is known as the biological window, where the near-infrared window resides. And as we know also, there's an inverse relationship between the length of, so as you go longer in wavelength, you can penetrate deeper into the tissue. And this allows us to interrogate signals 1 to 2 centimeters in depth. And finally, there are a lot of commercially available fluorescent probes for in vivo optical imaging, which make it ideal to specifically target this area. And then from the other side of things, instead of using a 2D planar technique, we have opted to put this all into a rotating gantry, similar to PET and SPECT and CT and MRI. And so using this optical tomographic technique, we can now introduce what FLECT is. So FLECT stands for Fluorescence Emission Computed Tomography. And it's this combination of using a rotating gantry accompanied by a ring of photodiodes. So I'll quickly walk through this diagram on the left. The black rectangles represent the photodiodes. The red rectangles represent the filters that are placed in front of the photodiodes. We have our subject here, and then when the animation starts, you'll see that the laser also is placed onto this rotating gantry. So everything rotates around the animal, and we were able to acquire multiple source positions relative to the position of the animal, while simultaneously collecting epi and transillumination fluorescence data. And so we acquire large amounts of data from this, as in one revolution, depending on how many source points you have, you can gain thousands to tens of thousands to hundreds of thousands of data points in one single acquisition slice, which is 1 millimeter. We then move the animal on a transverse bed in the transverse direction so that we can acquire over this entire range over the body of the animal. In addition to this, both the CT, the X-ray CT, and the optical components are mated onto the same gantry. I also need to reiterate that acquisition is done one at a time, but meaning one sequence of data is taken at a time instead of both simultaneously. But this allows us to view the animal from anatomic reference, getting a nice X-ray CT, and then interrogating over the specific area of where you want to look at for your optical signal. So we then acquire all this data, and then we render this into a 3D projection with the associated tomographic planes. So we use a 3D finite difference model for light propagation within the reconstruction algorithm. And this accounts for optical properties of both absorption and scattering and differences based on the tissue type. So depending where you are on the body, you can assign these optical properties accordingly. Like there's a section that we recommend for the head, another section that we recommend for the thorax, and the associated organs within the animal. And then we also utilize the CT data to enhance the reconstruction input parameters. And this allows us to give you the best model for simulating the light transport into the animal, in and out of the animal. In addition, we also use a 3D boundary scan to create a volume from the surface, or create a surface from the volume. And then finally, we use a DICOM format for visualization. So you can use whatever program that you like. Specifically, our system comes with vivo Quant, which is from in vitro, but you could use things like Ameris or ImageJ. And as I mentioned, data is acquired in slices, then reconstructed into a 3D stack, and then finally processed for visualization and analysis. And so we really are trying to move from this idea of a 2D image where you can see on the left to 3D tomographic images, so we can see on the right. And we can get a nice fast screening method from a 2D image without depth information and limited 2D localization just in the XY plane. But with 3D, you can now visualize your data in 3D. You can localize it with respect to the anatomic reference, and you can get an exact depth information from your data. And as something that's an interesting note about this particular study, which we'll go into more details in the application section, they were able to correlate the signal the volume of the signal that was reconstructed in the 3D data much better than they were able to see on the 2D data, which they acquired on the left using a planar imaging instrument. And you also notice that there is conveniently a hole cut out in the neck of the animal due to the fact that because of the strength of the signal, they were unable to actually visualize it without cutting it open. So they had to sacrifice the animal to do so. But we'll go into some more of these details of this study later on. So this idea of, again, continuing this train of thought, 2D to 3D, you can see these 2D methodologies, which are highly prevalent, bioluminescence, 2D fluorescence, to a limited angle 3D, which is still based off of this planar imaging geometry, tomosynthesis, and then finally to our format, which is 3D fluorescence tomography. And now we'll talk about the system. So this is the standard configuration. There are 4 lasers, 642, 705, 730, and 780. And we have associated fluorescence emission filter sets highlighted by the center wavelength and the full width half max of each filter. So as you can see, they're centered around 700, 800, and 850. The instrument, as you can see on the right, is a benchtop instrument. It can fit into a lab that has space on a benchtop. There are no special permits that are required. Even though we have an X-ray CT, it is fully shielded. And we also have these nice X-ray filters that we've installed that can reduce beam hardening and also apply to very specific bone imaging and tissue contrast imaging applications. And so this is what we hope to present to you guys, is that data from our customers has been taken and visualized in a 3D format. And what I really will highlight is how the study utilized the FLECT to gain information that otherwise would not have been possible had they used the 2D planar imaging or another technique. And so we'll be presenting these types of images from this point onwards. Maximum intensity projection will have a nice rotating GIF. And then the corresponding tomographic imaging views of the sagittal, coronal, and transverse. So starting off the applications, we actually have two key research partners here in the US. We work closely with the University of California, Irvine, the Functional Oncoimaging Department of Radiology, and also with Johns Hopkins, where they're doing some nice studies, which unfortunately were interrupted by COVID-19, but that's kind of besides the point, where they have a PET MR system literally next door. So being able to translate these optical probes and radioisotope tracers is the main focus at Johns Hopkins. And later on this year, we hope to be able to talk about their data in a meaningful way. So first start with this cardiovascular research. And this is actually from our customer in Australia. And what they did was they devised this, an antibody that in the presence of these activated platelets allows for detection of these arterial thromboses that they have created using a ferric chloride model. And so I'll go over the images on the top, on the left side. There's a mutant form where the antibody does not specifically target this ferric chloride induced thrombotic model. And then on the bottom, they have the activated version, or this targeted version, where it does, in the presence, the antibodies bind to these activated platelets as a result of this embolism. And so these antibodies have been conjugated with cyanide-7 and works perfectly with our system in that it's in the near-infrared region using excitation and emission. And then on the right, what we have is later on in their study, they used a control of PBS versus urokinase integrilin therapy to remove the blood clot or to remove the embolism, and then used their activated or their antibody conjugated to Cy7 in the presence of these activated platelets to detect whether or not the embolism was there anymore. And again, this was able to be performed on these mice without having to sacrifice them. So that is the main point here, and that their correlation between the visualized intensity was very well coincided with the volume of the actual embolism. And so without using FLECT, they would not have been able to see this, or they would have to have sacrificed at multiple time points in able to get this type of data. Similar application area using these labeled antibodies also is in the presence of cancer. So they performed a xenograft tumor model where they utilized their labeled antibodies, and again were able to specifically target and visualize where the tumor was in the hindquarters of this animal. And again, you can see the stark difference between visualizing a tumor volume versus seeing a 2D planar image where you just get a confirmation yes, no, without too much quantification or the ability to quantify how big that volume is. Another application in oncology is a study that we've done here at UC Irvine. And what we did was we injected some 4T1 mammary carcinoma cells, which have been then we injected angiostensin 750EX, which is a vascular targeting probe. and then allowed it to circulate. We took this particular image, I believe 24 hours after injection, and you can see that the probe accumulates quite nicely just in the tumor area of the animal, which you can see is in the bottom hindquarters. And this accumulation of the tumor and being able to quantify how large it is without having to cut or resect or measure with calipers is a very important tool that you can use using Insight FLECT CT. Another application area is in neuroinflammation. This comes from our customers in Latvia. And here they have an injection into the brain of 1 microgram of lipopolysaccharide and 5 microliters of ProSense 750. And here they induce this brain inflammation model, and then we're able to utilize ProSense 750, which has nice near-infrared properties, again, to visualize how much inflammation occurred over the area where they caused the inflammation at different time points. Next, we have our customer in China who used a previous version of our system without the X-ray CT, but essentially they have a breast cancer model where they have created a prodrug, a linker, and a fluorescent dye all conjugated onto the same molecules. And this linker is cleaved by the presence of glutathione. So using this breast cancer model, they were able to track the distribution of this drug dye conjugate in vivo. And in addition to this molecule having photosensitive-like properties, they are able to use the excitation of the fluorescence to actually enhance the tissue penetration of the actual conjugate into the breast cancer model. And so you can see in these maximum intensity projection where it is specifically in the body and how it has been nicely synthesized in that particular location. And here is another application area where we've, our customer in China has investigated the use of using VEGF and acting as a multifunctional probe, being able to conjugate both a radioisotope PET tracer and the optical fluorescent dye to the same molecule so that they can translate their findings from one tomographic method to another. And so here is a really good example, and here's something that we're going to be working with our collaborators at Johns Hopkins of doing a similar types of studies in different application areas where using an optical method can give you a lot of a priori or really good information before you move into a very expensive PETs or a PET radio tracer. Next application area is from our customer in Russia, where they designed a glioblastoma model and then created a theranostic conjugate. So this is the combination of therapy and diagnostics into the same chemistry. And there are several properties of this theranostic conjugate, including it's a chemotherapy MRI contrast agent. It also has a drug delivery agent, an albumin, and it also has a fluorescent dye conjugated to it. So these nice maximum intensity projections can show you the different imaging modalities and the ability of the FLECT CT to be overlaid on top of an MRI, which can give you all kinds of other important information that you would not normally get from either an anatomic or MRI-based method. Now we're going to move on to some orthotopic tumors and metastases, and we'll first start with this lung carcinoma model from our customers in France. And here they used RGD conjugated to Cy5.5 to target specific integrins in the presence of this lung cancer model, and we're able to visualize these small tumors inside the lungs. Similarly, they also used RGD and ATX in a colorectal cancer model to visualize these primary tumors, and they were able to use a 2D planar imaging system as they performed bioluminescence to actually correlate these two types of data. So both from the fluorescence imaging and the bioluminescence, they were able to see similar types of behavior and get more information from the optical imaging side. Next, we will talk about near-infrared fluorescent proteins, and these are gene reporters specific for different phenomena or behavior inside the body. So these reporter genes act like luciferase, but instead are tagged with near-infrared fluorescence. And you can transfect these into cell lines similar to bioluminescence. And the advantage is that you can get this you're able to see deep into the tissue. The fluorescence is localized only to the area of the cell accumulation, so you won't have this problem of non-specific targeting. And you can also minimize your background fluorescence because you know that the signal is only supposed to show up in one specific area. So for these, we actually used a commercially available 4T1 cell line that had already been transfected with IRFP and luciferase, and then we let the tumors grow into the body and image them over several different time points, showing day 10 here, nicely over the tumor volume. So to give you an idea of how you can also use transfection of an IRFP to, instead of a luciferase model, that's one possible way you could transition from bioluminescence to fluorescence imaging. And as Aaron mentioned, there's a new emerging application in virology where gene reporters have also been established as kind of the standard of care for doing this type of imaging. There's lots of traditional in vivo bioluminescence screening methods that are used. And in the latest work, we can see that these organs have to be excised so to perform a lot of ex vivo imaging, and that there's a clear application area for new research in this area for 3D tomographic optical imaging using our instrument. And so the answers now back to this quiz that we gave in the middle of the presentation. We start with PET in number 1, magnetic particle imaging in number 2, 3D fluorescence imaging from our instrument, SPECT imaging, MRI, and finally bioluminescence. And hopefully you guys got this right. I'd like to conclude by saying thank you for your time, and be happy to take any questions.
Thanks, Wesley. That was terrific. I'll go through and unmute everyone so you can actually ask questions. But while I'm doing that, I've got a question to start with. You mentioned IRFP before. Is there a better sensitivity with using that sort of approach as to injecting a probe in for, say, labeling tumors?
It's a great question, Steve. What we have seen is they can work equally well. The difference is the specificity, meaning like where the targeting agent can go. And a lot of times in research, that's a big problem to overcome. So if you're able to transfect the IRFP directly to the cell line that you're trying to interrogate, then you can specifically only get signals from those cells and not in other parts of the body, as opposed to injecting a fluorescent dye conjugated to something.
Yeah.
Can I ask a question?
Questions?
I know most of the fluorescent dye can be photo bleaching. So you use this near IR fluorescence dye. I also used it before. It's quite easy to photo bleaching. So how do you solve this problems for your detection measure?
So if I can understand the question, you asked, how do we combat photo bleaching, or in cases where there is either too much signal or signal that has caused a quenching effect? Yeah. Did I answer the question? So that is a great question. I think we would have to work on the specific protocols with you to try to reduce the signal in that case. What we've seen from the different application areas that I've highlighted today, and also some of the studies that we've done at UC Irvine, are that we tried to quantify things on the minimize, the side where we can barely detect the signal. And that's where I think the instrument can highlight its capabilities. So yeah, specifically with photo bleaching, yeah, we can talk a little bit more offline about that.
And second question is, for example, you use the NearIR in the 700 nanometer between 800 nanometer as the excitation source. And this excitation also can be absorbed by water, even this absorption is lower, but if you use a high power to excite the mice or in vivo animals, then it will cause the temperature of the animal increase quite high, then might be damages the animals. So do you have any solution for this?
So we do not use high-powered lasers in our system. So it's essentially a very small spot that diffuses into the animal. So
what's the power density?
I don't have the number off the top of my head right now, but we have performed this study in the past, so I can get you that data. I can assure you that it's not something that raises the temperature of the mouse significantly that causes that kind of thermal damage.
That sounds quite good.
Listen, and I think there's a point to say, which is the method of acquisition, Wesley, right, which is thin tomographic slices as opposed to the whole mouse being illuminated at one time with a camera being used to acquire the image, right?
Yes. So we're not illuminating the entire field of view. We're only illuminating one spot on the surface of the animal that then diffuses in the light that then diffuses into the animal. If that makes sense. As opposed to, like Aaron mentioned, illuminating the entire animal and trying to acquire everything at once.
Okay.
And I know with as a follow-up, Shanlin, when you mentioned that you have up-converting nanoparticles, I said that the standard configuration for the system is those 4 lasers, but we also have options to have non-standard lasers as well.
Okay, that's perfect.
Are there any other questions? If you want to ask a question, just unmute yourself and speak up, please. Appears not, Wesley.
Hi, Wesley, it's Mike from Monash.
Mike.
Have you guys made any further work on using the CT data to try and correct for scatter or attenuation or anything like that? Similar to what you would do with
and that is in the next release of the software, which should be coming to you guys pretty soon.
To explain to everyone, there is a current generation instrument at Monash University, at Monash Institute for Pharmaceutical Sciences. So Mike DeVere and I think Cam Knoll is attending this talk as well. But if you want to get in touch with me, I can put you in touch with those guys about getting access to that instrumentation.
So yeah, just to be clear, there is an instrument in Australia right now.
Yes, there is.
So, and that's where currently.
So, I guess the other question I've got for you, Wesley, is around COVID. Have you guys had any? I know you showed the viral construct work at the end there. Have you got any other applications you can see, immediately that could be applied with for COVID-19 research?
So that's a great question, Mike. We actually just over the weekend moved our instrument in China to a BSL-3 laboratory at the Academy of Sciences in Beijing, and they are going to be doing some COVID-19 research on it very shortly. So as that data comes out, we will definitely make it available with their permission. I know a lot of current research is looking at changes in lung structure or lung damage that occurs from COVID-19 models. And so I think the CT is a very powerful tool in that case.
Yeah, even the CT on its own is good for that. Yeah, no, I was just more thinking about immunology applications as well. But,
yeah, Michael, it's Aaron here. It's a bugbear of mine because it's there was a WMIC event a few weeks ago talking about best practices in COVID imaging, and it was purely PET centric, as you would expect, because PET's the standard everybody has. There was a passing reference to those couple of studies we highlighted, which are seen as the most advanced optical imaging studies, which are, they're not that different, as you say, from, immuno-oncology or any other oncology studies where you're just looking at rough patches of bioluminescence and it's, it's there, not there, big, small, growing, shrinking. And even our friends at PerkinElmer published a bunch of longer studies. I've been pushing them to see if there's more stuff that they can help us with. And, but it's the same stuff over and over again. And it's just firmly rooted in this idea that, if you can't see inside the lungs easily, why bother? So researchers just really haven't bothered. So I'm hoping that whether it be at, whether it be in the Chinese Academy of Medical Sciences or a couple of other places, we're working on grant applications right now. Here in the States for a couple of places where they've been repurposed from immuno-oncology into COVID work.
Yeah.
Seeing the virus with transfection is nice and easy, and then using similar techniques to immuno-oncology to follow the immune system around with conventional tracers, and then unmixing the two wavelengths should produce some really nice images, to help people along in the virology work without having to administer PET scans, which is, it's not what you do in a human, right? So if you can avoid doing it in the mouse, it should be more realistic.
Yeah, that's kind of where I was thinking. I guess the other question on immuno-oncology, has anyone done this with CAR T cells with the infrared proteins?
Yeah, that's another emerging area, and it would be really nice if we could have some examples on our system. That's something that we're definitely exploring right now too.
Wesley, you've done some studies recently exploring the sensitivity of the instrument, where you label some cells and then were able to detect them. Can you comment on the levels of sensitivity you were able to achieve?
Yeah, so what we do with each instrument is we have a calibration curve that's performed where we have varying amounts of fluorescence activity in known volumes and concentrations, and then we perform a serial dilution down to the sensitivity limit of the system. And that gives us an idea of what the system performance is and how it can be applied to specific concentrations inside an animal. And with some recent pilot studies that we performed at UC Irvine, we were able to see down into the hundreds of cells using a dendritic cell application that have been labeled with ICG near the surface of the animal. So I will say things in such a way that it really depends on how much fluorescent signal you can get, and then whether or not that's also specific versus something that is untargeted, and also where it is in the body.
But effectively, you're able to just detect hundreds of cells, not
correct.
Not with thousands. That's pretty spectacular, I would suggest. Christoph, do you have any questions? Or Karen?
No, not at the moment.
No, not at the moment. I'm pretty fine. That was excellent. Thank you a lot.
Yeah. Well, if there's no more questions, I'd just like to thank everyone for attending our our webinar, and thank Aaron and particularly Wes for giving a fantastic presentation. So thanks, guys. And again, any questions or further things you'd like to follow up, please get in touch with me. You should all have my email from the correspondence, and I'll be happy to help or put you in touch with these guys. Thank you.
No worries.